Okay, so this is the typical Punnett square, where mom is ducks and dad are beavers, and that's how we got the platypus. No, that's not how we got it. Right, but I'm still curious about how the platypus came about. Put a beaver and put a beak on it, make it swim. Make it lay eggs, but make the eggs leathery like lizards lay eggs. I don't know. I don't know what God was thinking about it, but anyway, it's a creation. All right, so let's talk a little bit about genetics. This is the last topic of the whole thing. It's pretty big. It incorporates 2 chapters in your book, but I condensed them into 1 PowerPoint. Okay? So that's why the PowerPoint looks a little bit big on it. All right, so let's talk about what a gene is, right? We've been talking about genes. It's the unit of heredity that encodes information to make proteins, cells, basically the entire organism. A gene, for our purposes, is a trait. Okay? You've got eye color. That's a trait. That's a gene. You've got different gene types. We call them alleles. We'll get into that, but then you get blue eye color, red eye color, but the gene is eye color, right? So we have something called a locus, or more than one locus. We refer to it as loci, and it's a site where on the gene, where a gene is actually located. So we actually discovered this. It's called the genome, right? The genome is knowing where every gene is on the whole DNA molecule, where every locus is for those genes. It took 10, 15 years to actually do that. It's called the Genome Project. So we have inheritance, and this is the process where the traits are passed down from offspring to offspring to offspring new generations. Okay? So just defining some words here. All right, so like I said, we can't talk about genetics without even mentioning this guy, Gregor Mendel. Right, which was this guy over here. He's an Austrian monk, or he was, okay? And he established basically the rules of inheritance in 1863. He worked with a lot of pea plants. So you can imagine all the names that he was called after that. He was working with someplace in there with his little pea plants and stuff, realizing that this guy's a genius. Okay? So what happened was he did these experiments in the monastery garden with these pea plants. Okay? The edible pea plant. And he saw patterns as he was doing this. I always want to get into his head and say, where did he come up? Where does this curiosity come? He's just looking at a pea plant and realizing there's different colors and you want to do experiments. I mean, that's drawing a hypothesis, right? You observe things and draw this hypothesis. So he saw patterns in a way that plant characteristics were inherited. Now, when we get into a little bit later, there's going to be a lot of things that follow his rules that he actually discovered. And then there's things that just are not, we call them non-Mendelian genetics. That don't follow the rules. But if he didn't come up with this, we would have never found out other things that are non-Mendelian. So he's really the father of all genetics. Okay? So, and he also employed statistical analysis. He did things where he could actually, if he did, let's say, 100 plants or 200 plants, he actually saw that maybe 25 of them were green and another color was over here. So he actually put numbers to this and started realizing that things are changing after generation to generation. Well, why? Why does this happen? Okay? So why was he successful with doing this? Well, he chose the right organism with which to work with. Okay? Designing and performing the experiment correctly is key to this. And he made a lot of notes, and it made sense. Analyzing the data properly. Okay? And that's what he did. He chose the right organism because he has complete control. He's not killing off anything. He's not using rats or anything like that. He's killing off things. And the process is very fast. You don't, it's not like you do it to, let's say, a rat, and you got to wait for the rat to deliver a baby or a baby rat, and that might take weeks. This happens relatively fast. So here's the advantages of using these pea plants, picking the right organism, all right? There's a lot of hereditary features, all right? There's color, and we can also talk about if it's wrinkled or not. We can talk about the leaves, what they're doing. There's a lot of different things you do here, all right? So there's a lot of variants, meaning a lot of traits, purple and white flowers themselves. The mating plants can be controlled, all right? Think about it. If you had the two rats over here, say the one rat, okay, she's okay, go over there and mate with her, I mean, it's hard to do. This is easy to control, and I'll show you what that's all about, okay? Each pea plant has both male and female carps. So we have the stamens and the carpels, right? The stamens are basically the sperm-producing ones, and the carpels have the egg-producing. But each one of these plants have that, so you could actually do things, all right? You can do this cross-pollination. It's very easy. Fertilization, you're just going to take some stamens from one and just bring them over to the other. It's very tedious because it's small, but it's very controllable, okay? You just dust one plant with pollen from another. Just take a brush, like a little paintbrush, and just put the dust in another plant. So just showing you what it is, this is what the pea flower looks like, but when you look inside, you're going to see the carpels, which is this green little thing over here, and then you've got the stamens over there, which is the male producing thing. I won't ask you about those, but just to give you an idea of how this was easy for him to work with. Okay, so he started doing, the first thing he was doing was this monohybrid crossing. So this is his experiment when we're just dealing with pea flower color only. We're going to talk about other things, other factors later on, but let's just deal with just the color itself. Okay, and that's what he did first. So you have this crossing or this mating, let's call crossing and mating, same thing, of two true breeding varieties. So what you have here, notice it didn't show well, you basically have a purple flower here and a white flower over here. And these are true breeding. We've been, to make a long story short, he followed these two plants, realizing that when he did all this, this one was only producing purple flowers, and this was only producing white flowers for like probably 10 or 20 different generations. So he wasn't so concerned. This is like true purple, and this is true white. But then he crossed them. What happens if you take a purple flower and a white flower? What do you get? And what he ended up getting was 100% purple flowers. We call that the first generation, and it's referred to with F subscript 1. So that's his first observation. You take a purple and white flower, and you get all purple flowers. Okay? So he did self-pollination. In other words, he took two of those purple flowers that came out of there, and he mated those, across those, creating our second generation. Does that make sense? Are you following with that? When he did that, he noticed that 3/4 was purple, 75% were purple, but 25% were white. So now he got a little bit confused, because where did that white come from? It didn't show up at all in that first generation, but the second generation there was. So he had some thoughts with this. He allowed the first generation to self-fertilize, the purples, to cross. And the second generation had a 3-to-1 ratio, 75% purple, 25% white. the white trait didn't disappear in the first generation. It was hidden. And it got passed on in the second generation. That's pretty neat if you think about it. Talking about plants, right? Something, it wasn't there, it was hidden. So this was the experiment. He took purple flowers and white flowers. All the plants had purple flowers. He then took 2 of these purple flowers, he bred them, and he got 705 of these and 224 of these. Basically, 75% purple, 25% white. So he's put in this statistical analysis. He didn't just do 3 flowers, he did friggin' 1,000 flowers. That's a lot. That's not a small sample. And I'm sure he did it with, let's say, 10 or 20 other experiments and was producing the same percentages. Interesting. He then self-fertilized the second generation ones. All right? So you have this, are you following? The third generation now. All the white flower plants produce white flower offspring. All of them, 100%. These prove to be true breeding. In other words, they are white. There's no purple in them. So in the third generation, the purple flowers from the second generation, they produce 2 types. 33% were true breeding purple flowers. So he looked at these and he did other experiments and said these are only, after generations, they were only putting out purple flowers. But he also noticed that there's 66% of those were hybrids. They actually have both purple and white flower offspring. Again, in the ratio of 3 purple to 1 white. So therefore, the second generation had to include 25% true breeding purple, 50% hybrid, and 25% true white breeding. Does that make sense? You're following all that. It gets confusing. We just came in right now, which no one did. You're probably saying WTF. Right? So you have to just follow that. So he reasoned that the only purple flower factor was affecting the flower color in the first generation hybrids. Okay? We call the purple flower something called a dominant trait. Because that's the one, if you had it, it shows. Even if there was a white trait in there, the dominant trait shows. But the white flower was a recessive one. All the white flowers were true white flowers. They didn't have any purple. We saw the same pattern in 6 other pea plant characteristics. All right? Representing 2 traits. So what Mendel did was that he called it a hereditable factor, which now we call a gene. So I'm saying he's the father of genetics. He put all these together. So he did this without even knowing the concept of chromosomes that happened. They discovered chromosomes 20 years after what he did. So he really set the spark in the bucket so that other people wanted to learn more about what was going on. All right? Meiosis chromosomes wasn't even heard of at that time. So Mendel put laws together. And we have Mendel's law of segregation. And what it is that we have this thing called an allele. An allele is, and I have it written in some place to find a little bit later, but an allele is a certain variant of a gene. In other words, a gene is eye color. An allele is blue eye color. An allele is brown eye color. So they're variants of a gene. Does that make sense? Okay? Flower color is the gene. Purple and white are alleles. So allelic pairs segregate or separate during gamete formation and then randomly unite in fertilization. So an organism has two alleles for one trait. In this case, the color, right? These two alleles separate, now we know, during meiosis. And a sperm and egg carry out only one allele for each inheritable trait. So you're going to get one allele from mom and one allele from dad. Which one actually shows up on you, that's where he put it in. If you've got a purple skin color from mom and a white skin color from dad, what color are you going to be? Purple or white, right? But if you carry purple, thing is, are you a true purple or do you have what you need to? Does that make sense?
So I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but they can be really hard to find. And while I doubt they'd do so well in the wild, their genetics are fascinating, and so are guinea pigs. We spend quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene, and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has two recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter, or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait, not having hair, shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase h. A guinea pig that does have hair can be represented as a big H, or a big H. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h, is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is a lowercase. If I were to show you a guinea pig with hair, would you know its genotype? Well, you would know it's not little h, because if it was little h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H. We don't really know. I mean, we could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know for sure that it's little h, because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means one because it focuses on one trait, in this case, hair. And that means a Punnett square would need to be created like this with 4 squares. In our cross, we are going to cross 2 heterozygous guinea pigs. So let's look at this in steps. Step 1, first, figure out the genotypes of the parents. Heterozygous means big H, little h. Step 2, place one parent along the top of the Punnett square, outside of the boxes like this, and place the other parent along the left side of the square, outside of the boxes like this. Step 3, cross them. For formatting purposes, we always put the capital letter first. The results that you get in the squares would be the offspring, the babies. Now, if I were to ask you the genotypes of the babies, you could list them out. 1 big H, 2 big H, and 1 little h. I could even turn that into a genotype ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Or a percentage. 25% are homozygous dominant, 50% are heterozygous, and 25% are homozygous recessive. Well, what if I asked about the phenotypes? What does that mean? I like to think that pheno sounds like physical. So the phenotype would be the physical traits of that organism. And in this example, it's whether they have hair or not. Now remember, any babies that have a capital H have a dominating allele, and they will have hair. So the babies that are big H, or big H, they have hair. So 3 of them here, the 1 big H baby and the 2 big H babies, they all have hair. The homozygous recessive baby, which was little h, that baby has no dominant allele present. So this little guy would be hairless. So you could say the phenotype ratio is 3 have hair to 1 that's hairless. Or a phenotype percentage could be 75% hair, 25% hairless. One thing to emphasize about Punnett squares is that they are predictions. These are probabilities. This means it's not necessarily exactly what you're going to get. For example, it's a probability that a child has a 50-50 chance of being born a boy or a girl. But we all know families that only have children that are girls or families that only have children that are boys. Probabilities are predictions. Another reason to love biology, it's exciting. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.